honored to introduce the next distinguished speaker, Professor Vinetz from UC San Diego School of Medicine. Clinical training at the Johns Hopkins and research training at the National Institute of Health. So he took a faculty position at the UTMB and then returned to UC San Diego 4 years ago. So the presentation of Professor Vinetz is entitled Prospek for Malaria Vaccine.
I'm going to talk about prospects for a malaria vaccine. And from one title, there's going to be two completely different answers. This young man represents the problem with malaria in the world. He has malaria parasites in his blood. He lives in the Peruvian Amazon region of Iquitos. He is asymptomatically parasitemic. In fact, he's annoyed because he was going to go off and play a soccer game, and we asked him to come to the lab. So that's the definition of an asymptomatic patient that has malaria parasitemia. And the focus really for eradicating malaria is on this guy and everything that he represents. And so I want people to keep that in mind during my talk. Okay? So the malaria problem is that there are billions at risk, 500 million infected with malaria every year. But here is the key thing. So everybody throws out these numbers, but when you really parse them, about 20% of these 500 million are actually clinically ill. And so that means 400 million or so are not. That's the reservoir of malaria. That's the ecology of malaria in the world. This is the problem, very baldly stated, of malaria eradication. Okay. So Ronald Ross discovered he was a physician scientist living in India, actually used a bird model of malaria transmission and found out that mosquitoes transmit malaria. And because of that, he actually led and contributed in a major way towards the only successful way that malaria control efforts have ever taken place, and that's through vector control. And we've already seen some information about that this morning of the potential benefits and pitfalls of that. But this was really led by Ronald Ross. By the way, he was a terrible poet. Do not read his poetry. Okay. So I'm going to talk about this complicated thing called the malaria parasite and its life cycle. And what I'm going to do is use that to organize my talk. The talk, when you talk about a malaria vaccine, actually has to do in some details about the parasite life cycle. So as we know, Anopheles mosquitoes inject the sporozoites into a person. You have a liver stage here. The liver stage leads to the blood stage, which is actually where the disease takes place. And then a very small proportion of the red cell invading parasites will differentiate into boys and girls, which become young men and women in the gut of a mosquito and actually have sex fertilized, and that's where genetic recombination takes place. And in fact, the mosquito is the definitive host of the malaria parasite. So these are the key cycle stages, and I'm going to refer to those through this talk in very specific ways. Okay, so to put that life cycle into a different context, what's the purpose of a malaria vaccine? Well, it's obvious. We want to do what we do for measles and mumps and rubella and maybe Haemophilus influenza and other sorts of vaccines that really work. That means get rid of the disease and that you vaccinate somebody and they don't get it. So that's that's what we all think about a vaccine. And in malaria, it doesn't work that way. And you have to completely think differently about malaria. So if you want to prevent people from getting infected, you have to target the sporozoite. Again, Dr. Kampa is going to talk about that, and I'll talk about that a little bit. Another thing is, well, even if it's inevitable that people are going to get infected with malaria parasites, at least can we try to ameliorate the severity of disease? Can we make people less sick? Can we prevent pregnant women from getting complications in pregnancy? Can we prevent severe anemia in children? So we want to reduce the disease severity. So that's not really a prevent the infection, it's to prevent the badness of the infection, if you will. And finally, there's another approach, which is just to prevent transmission to begin with. If you, realizing that humans are the only reservoirs for malaria transmission to mosquitoes, if you get rid of the reservoir or you prevent that reservoir from infecting the mosquito, you don't have to go and do all the vector control stuff. At the vector level, you prevent the mosquitoes from getting infected. This, it turns out, is the least popular approach and has, until extremely recently, let's say over the past year or so, has not seriously been considered by the major sort of decision makers in malaria vaccine development. But then the E word came about a year ago. And the word eradication means if you're going to try to eradicate malaria, you got to get rid of transmission. It's not good enough to reduce disease severity, and it's not good enough to have a partially effective sporozoite vaccine. If you want to eradicate it, you got to prevent. It's like, and the way I'll put it, and maybe I'll even say this metaphor again, wrapping up, it's like if you consider wrapping every person in a protective shield during the entire time that the malaria parasite's in their body, and realizing that most of the people with malaria parasites actually aren't sick, you prevent them from passing it on. Those are the ways that you have to think about any set of malaria vaccines, and that's something that's really very different than normal vaccine considerations. And so, if one is a vaccinologist, and I am not yet a vaccinologist, but actually that's something I want to do when I grow up, because I want to take the science I do in the lab, and I actually want to do something with it. I want to do some translational work. And I'll show you a little bit about how we're doing that. But if you're going to go and vaccinate somebody, you want to know, well, we can't challenge people, so we have to have a surrogate marker of protection. So, for example, with measles, you vaccinate somebody. If they have titers of antibodies, you say they're protected. Same thing for hepatitis A or hepatitis B or Haemophilus or whatever. The problem in malaria is that if you vaccinate people, you don't know if they're actually protected because we can't challenge them. So then you must do properly powered, relatively large field studies of malaria. And then there's all the complications of the human subjects, and then people have already had malaria. It's never a pure system. So this problem that we can't actually predict protective immunity in the laboratory is a major issue. So this has to do with each of these stages of malaria. So we do not really know how people become immune to sporozoites or blood stage parasites. We know that people walk around and they're perfectly clinically immune to malaria and they got parasites in their blood. That means their immune systems are controlling their parasitemia, which means they have a protective immune response, but they don't have sterilizing immunity. So they're immune, but not perfectly sterilizingly immune. It's hard to study rodent malarias to infer protective mechanisms in humans because they really are very, very different biologies. You can get some hints, but it still doesn't give you the answer you need for doing vaccine testing. The only exception to that actually is in transmission blocking immunity, and I'll talk a little bit about that. Whereas if you can actually measure levels of antibodies and if they protect mosquitoes from infection in the laboratory, you know it's going to work. So with that one exception, we don't even have laboratory correlates of immunity. So let's start with the first of the three stages of malaria vaccinology, and that is an anti-sporozoite vaccine. So this Anopheles mosquito is injecting during blood feeding sporozoites, and within usually minutes, but certainly less than half an hour, a single sporozoite's inside of a single hepatocyte. And the idea is to interrupt it right there. So is it possible? Dr. Kappa, again, is going to talk in more detail. But this goes back to one of his mentors and really the leader. This is from Ruth Nussenzweig's paper, and so you can see there she is up here. This is published in 1967, where she took rodent malaria parasite called Plasmodium berghii, and she infected mosquitoes with the parasite and then buzzed them with some radiation. And it turned out that these irradiated sporozoite and vaccinated animals were actually partially protected, variably protected from 25 to 88%. So this said, you can actually use an attenuated sporozoite to actually help protect an animal against the primary infection from malaria. So all of work has actually proceeded from this point. In 1984, and I don't mean any disrespect to the NYU group, but the NIH group and the NYU group at the same time published the sequence of the first malaria parasite gene ever cloned, which is circumsporozoite protein. This came from the NIH lab, and this is just the whole genome sequence. There'll be a quiz at the end of my talk. But the point is that this is in 1984. And right now, the clinical trials are actually continuing to go on based upon this protein. And only in the last 10 years, and really only in the last 6 to 8 years, has the funding and the practicalities of doing such a vaccine trial come along. These things take a long time. One of the trials based on something called RTSS, which is a recombinant protein with the hepatitis B vaccine antigen fused to the circumsporozoite protein with an adjuvant, was given to young African children in Mozambique in a randomized controlled trial. And the numbers, this is one of those Rorschach tests. It's either the glass half full or the glass is half empty. I can leave it to you to decide. Protection was maybe anywhere from 50 to 60% in the short term, and this waned. And at the start of the following malaria season, maybe a quarter of children were actually protected from a malaria, the first onset of a malaria episode. There was some protection that was relatively short-lived against severe disease. And this is the most advanced vaccine candidate. Again, against the sporozoite protein, this has been in the works for since really, this is the culmination of the irradiated sporozoite vaccine. The other alternative way of doing this has been led by this actually young-looking Steve Hoffman when he was still in the Navy, who, to cut a very long story short, has started a company to actually produce, in a clinical-grade way, irradiated sporozoites of Plasmodium falciparum in an industrialized fashion in sterile mosquitoes in huge amounts, and they're frozen, to actually deploy them to malaria-endemic regions. This is extremely audacious, and one can talk for a long time about this. And in fact, there have been people that have been vaccinated with irradiated sporozoites. We're talking with many immunizing bites from mosquitoes, and that there's actually a surprisingly good protection rate, and that the protection actually lasts a fairly long time. It wanes with time. It seems that this is a better way of vaccinating than a recombinant protein. The question is what's the basis of the protective immune response? And more importantly, if we don't understand that basis, how can we actually optimize the vaccine? These are still the open questions. And this is from a rural village in Mali. It looks kind of like a National Geographic picture. Greg Lanzaro at UC Davis gave me this picture. Every single person in this picture has malaria parasites in their blood. Every single time they get by an infected mosquito, they're going to get an infection, and it's going to be a productive infection. And we don't know that we can even develop natural immunity to regular sporozoites. The question is how to actually enhance that. This is, in fact, I believe personally that people in hyperendemic areas such as this actually develop immune tolerance to sporozoites, and the vaccinologists actually haven't considered this yet either. Some have, and many have not. And so this is, I think, one of the major impediments to an anti-sporozoite vaccine. Okay. So the pipeline, which comes off the PATH MVI Malaria Vaccine Initiative website, includes both the RTSS and the Malaria sporozoite vaccine and some other vaccines. I'm going to talk a little bit about these blood stage vaccines that are under active investigation. Malaria, again, is trying to make attenuated whole organism vaccines. And this is a nifty picture that Steve gave me. So somebody's dissecting out sterile mosquitoes because you have to be able to comply with the FDA regulations for a pure product that's not adulterated by bacteria. And their goal is to actually immunize specific groups of people, including infants in sub-Saharan Africa, pre-adolescent, early adolescent girls, and this is to prevent malaria in pregnancy. And then the whole thing would nominally be funded in the very preliminary business plan by people, travelers and soldiers, really, in the industrialized world that would actually pay for this vaccine, and this would then provide funds to optimally deploy this to the developing world. So this is it's a complicated and audacious approach to malaria vaccines. Okay. That's all I'm going to say about sporozoite vaccines. You'll have another 25 minutes or so of that. The next stage is against the blood stage, is against trying to ameliorate or lower the parasitemia and prevent the sequelae or prevent the complications of the disease called malaria. And Phil Felgner just showed this. It shows that great minds think alike. Nature now publishes all their papers online, so you can actually cut and paste them. You don't actually just have to Xerox them and scan them and take pictures anymore. And the bottom line is that one can use immune globulin from malaria immune adults and reduce the parasitemia. So that means that presumably there's protective mechanisms that, at least in part, are in the antibody component of blood that at least confers partial protection. They do this on a log scale, so we don't know if this is actually sterilizing immunity, and that's another issue. And in fact, the children that received this immune globulin way back in the 1950s actually developed gametocytes, so now they're still able to transmit. So you can knock the disease out, but you can still transmit, and so you've got problems. It turns out you can treat patients with chloroquine and do the same thing, or Fansidar, and you can actually get gametocytes very nicely as well. So drugs also need to be transmission blocking to be very effective, and that's a whole other set of stories. So there's a whole boatload of blood stage proteins. I liked Phil's analysis about how the top ones don't distinguish between protected and unprotected people, such as AME1 or MSP1, but EBA175 seems to be a protective antigen, and these are in various sort of clinical development, different adjuvants and different protocols, different countries, et cetera, different sponsors. So it turns out the furthest along any blood stage vaccine has gone really through phase 1 safety studies. The big problem, and I don't have time to talk about this today, is that these vaccines are really going to be aimed at preventing the mirazoite, which is released from a dead infected cell, from getting into a new cell. And it turns out that falciparum probably has a boatload, probably dozens of different ways of invading a red cell. And so you're going to have to, in order to make a blood stage vaccine, prevent all of the alternative pathways of invasion and trying to kill this evanescent mirazoite within the blood. This is going to be a huge challenge. And it's never going to lead to sterilizing immunity. It'll, at best, lead to partial immunity and to amelioration of symptoms. Important, yes, especially important in pregnancy. This vaccine approaches especially with, against proteins that allow the parasite to adhere to placenta. These are extremely important vaccine approaches. But again, these are all partial technologies, or what Lewis Thomas said, halfway technologies. Really getting at this underlying problem. It's really, in a sense, it's not going to do the eradication that we all want. It's a part-way technology, trying to at least do something. Okay, so again, I showed the pipeline. And again, the blood stage vaccine would be hopefully something that would protect people against symptomatic malaria. This woman's having a paroxysm of fever and rigor, or of cerebral malaria in children. Okay, so what I personally believe, and this is something that is a long symposium and a lot of beers and probably some knives and blood on the floor, is actually to prevent the transmission of malaria from people to mosquitoes. And that's again, antibodies directed at the parasite within the midgut of the mosquito. The idea is that you vaccinate people, whether or not they have malaria parasites, and if they do, and a mosquito bites them, then that person becomes a delivery device of antibodies to the mosquito that prevents the parasite from invading the mosquito. Okay? So it's a different way of thinking about a vaccine. We're trying to protect the mosquitoes of the world from transmitting it so that they don't transmit to people. People call this an altruistic vaccine. I think that's a very, very bad thing to call it. It's really a vaccine to induce herd immunity. And everybody here in this room has had at least a rubella vaccine. Every single man in the room never benefited from this rubella vaccine. What we use this vaccine for is to prevent pregnant women from passing rubella on in pregnancy. So we have herd immunity to prevent the circulation of this particular virus, and the Haemophilus vaccine as well has turned into an eradication of this bacteria from this country, even though it was only aimed at children between 9 months and 2 years of age, because adults don't get Haemophilus meningitis. So transmission blocking vaccine is not an altruistic vaccine. It is a herd immunity inducing vaccine. And if you talk to people in the developing world, they care about their communities, they care about their families, and they don't say, oh, if you vaccinate somebody else, that's going to help me. That's not a problem for them. In Europe, in the United States, if the vaccine doesn't help a person, they say, well, how could you do that? That's not ethical. And that's actually wrong. And in fact, it's wrong with more people in the world than it's right with. And those are actually the fighting words. So that's the argument. That's why there's blood on the floor with transmission blocking vaccines. So what's the evidence for it? So transmission blocking immunity was actually discovered in the same laboratory, actually published in 2 different journals, and I still haven't figured out the politics of this, but Bob Watts published in Science and Richard Carter published in Nature pretty much the same month, doing the same experiments independently. And what they showed was if you make gametes of Plasmodium gallinaceum, which is a chicken model, you can actually immunize chickens and prevent them from infecting mosquitoes. So these are purified macrogametes and microgametes. They were used to immunize the chickens. Let's say this is blood being fed to mosquitoes through a membrane feeder or directly from the chickens themselves. This happens to be human blood with Vivax in it being fed to Anopheles mosquitoes in the Amazon. Mosquitoes are dissected, and then you count these oocysts on the mosquito midgut at the end of the day. It's a very nice laboratory test. A little bit cumbersome. This led to the next series of experiments, which is if you can actually immunize animals to prevent transmission, how are we going to make that into a vaccine? 2 groups, the NIH group and the Nijmegen group from the early 1980s, when Plasmodium falciparum zygotes were used to immunize mice, make monoclonal antibodies, and then those antibodies were screened to react with the surface of malaria parasites. And so these are immunofluorescence images from the Nijmegen paper showing that monoclonal antibodies against the sexual stage zygotes actually bind to midgut-derived falciparum parasites. And if you can actually identify the antigen that those antibodies react to, you've got your vaccine candidate. And so David Kaslow followed up on that, and he was my mentor when I was at the NIH, and 20 years ago, 20 and a half years ago now, published the first vaccine candidate, PFS 25, which still hasn't gone into clinical trials. This is actually a clinical trial. They've, to cut a very long story short, Lou Miller's and the malaria vaccine group, John Robbins and Rachel Schneerson at the Child Health Institute at NIH have finally, and Yamin Wu, and a number of other people have actually provided a formulation and chemical modification of this protein that makes it immunogenic, long-lasting, and sustained rising antibody titers, at least in an animal model, and now an IND is being filed in a clinical trial, hopefully in Peru, and I'll show you the site, will actually be taking place. So this is a way of actually, it's a measurable immune response. We know we have the surrogate of protection, all based upon this more than 20-year-old protein. So the mechanisms of transmission blocking immunity are always antibody dependent, plus or minus complement. And of course, if you consider, and I don't have time to talk about this, but a mosquito is trying to digest its blood meal. A female mosquito bites people to get blood to lay eggs. So you would think, well, how could antibodies possibly last in a mosquito midgut while it's being digested? And it turns out they do, and it works. And so it depends on how long these antibodies last and the quality of these antibodies and a number of other immunological parameters. This is South America. Here's Brazil over here, and here's Peru, and you fly into Lima. Here are the Andes Mountains, and you fly over the Andes an hour and a half, and you get to the capital of the Peruvian Amazon city called Iquitos. In the Peruvian Amazon, it turns out that this was a place in the mid-1990s where there was a progressively increasing epidemic of both a falciparum and Vivax malaria, and this is really a late consequence of the cessation of the DDT and malaria eradication campaigns. And so one can see, before 1992, there was no falciparum malaria at all, and occasional sporadic cases of Plasmodium vivax. And then over the 1990s, one can see that there's really a rising epidemic curve, which is now partially subsided, but there's still tens of thousands of cases of both falciparum and vivax in the Amazon. What's really remarkable is that last year there were 3 deaths total reported from falciparum malaria, despite thousands and thousands of cases. So this is really remarkable, and this is why it's actually ethical to do a transmission-blocking vaccine trial in a place where you don't have to directly help somebody with an intervention. You can actually try to investigate the pure effect of actually trying to block transmission. Because can you imagine going to a Malian or a Gambian or a Kenyan village where people are actually dying from malaria and say, we're giving you a vaccine, but it's not going to help you right now? That's going to be a big problem until the proof of principle really is there, until probably there's a vaccine partner. Anyway, so this is a Google Earth image of Iquitos. It's a city of about 400,000 people. This is where the Amazon River forms from the juncture of several feeding rivers. There are rural areas out in the jungle, and this is an urban area. All the malaria is actually outside the city of Iquitos. We actually are working in one particular, several places, but one of real interest, and this is actually a global lesson for malaria control. So if you go from Iquitos and you go take a speedboat, about 40 minutes with a high-speed speedboat, you go down to a village called Masan. And Masan is actually where the Masan River comes down and meets the Amazon, and the Napo River comes a little bit further upstream. This is an important place because it's a central place where many people who are traveling and doing economic activities of various sort, fish farming, logging, etc., come and pass through. But even more importantly, people that live in all sorts of areas go up these rivers to do their economic activity. This is a typical looking, this is from a place called Sunguracocha, but this is a typical looking health post. This is one of our study sites with a 3,700 or so population. It's relatively young, and we're doing investigation of households. This actual economic activity that brings people from all sorts of different places, and it turns out they're mixing and matching their malaria away from the villages, and then they go back home to their home villages where they have permanent homes. This, as far as I can tell, this epidemiology of malaria transmission has not really been focused on or studied. And it's unbelievably important because it turns out that in this village, we can't even find any of the Anopheles vectors. We have thousands of cases a year in this region. So how can you have so much malaria if there aren't any vectors? And the reason is because many people go away and come back. So in a sense, it's local, micro-regional travelers' malaria. How can you possibly eradicate malaria under those circumstances? You better know how people are getting it, where they're getting it, and the only way of actually eradicating it is drawing a circle around a region, maybe this region, and vaccinating people with a transmission-blocking vaccine. So that's what we're actually proposing to do in this long-term vaccine study. And there's about 16,000 people in that region, so it's not so crazy. And what could the potential effect be? Potential effect, so this is from Richard Carter. He published this in the International Journal of Parasitology a few years ago. And actually, it's a PowerPoint presentation, but it's really elegant. And so I'm going to show it real quick. If this is a theoretical village with a perimeter of where malaria is transmitted and where a perimeter where vectors are actually breeding. In red, these are malaria patients, and in green are non-infected people. And so if there's a transmission blocking vaccine with 99% coverage, and this is within a range of 1 kilometer, so that's the assumption. Then after one round of transmission, which is a few weeks, according to the biology of the parasite, you don't see much of an effect, but at the end of 2 rounds of theoretical transmission, you can actually eradicate the transmission of malaria from that village. Falciparum does not live in people probably for more than a few months. Plasmodium vivax can live in people for probably, at the most, a couple of years because it forms dormant forms in the liver. And those are the most important forms. So if you can have a vaccine that has this is, of course, nearly perfect coverage and efficacy. But if you have partially effective vaccines and one can model these numbers, then you can actually see where eradication can actually take place, at least with a pure model of a transmission blocking vaccine. Now, this doesn't take into account drug treatment, secular trends, public health efforts, education, etc. This is just the pure theoretical effect of a vaccine. It's crazy. It actually looks like it might be feasible. And the main thing is, can you find the people to vaccinate? Okay, so that's where the transmission blocking vaccine comes from. I want to end up with a sobering thought, and this is, I think, the 3rd time today that somebody mentioned malaria in the United States. Does anybody know when it was actually eradicated? It wasn't until after World War II. And can anybody tell me the one reason that was probably eradicated? It had to do with the Tennessee Valley Authority, which actually was a work project to create electricity in dams throughout that part of the country. And it took secular trends, electricity, television, screens on windows, and general elevation of the living standards that actually led to the final eradication of malaria. So we can talk about vaccines. Now, vaccines are not going to necessarily do that. That's a magic bullet. Because, boy, is it hard. I think it's much harder to give everybody electricity than to give everybody a vaccine. Okay? That's the problem, and that's a sobering thought. And we need to be a bit circumspect about eradication in the absence of social justice and really the secular trends of social improvement. We have anophelism without malaria in this part of the world. We have Anopheles mosquitoes. So this is always going to be a threat everywhere in the world, short of the eradication. And that's the sobering news, and that's why if you think that one person can go and lead to explosive epidemics, when one realizes that, that's why eradication really is that much more important. And I'm simply one of many people that works on malaria in this particular very complicated group. We've collaborated in a whole series of investigations with this large group of folks, and it's all been supported really primarily by the NIH and a couple other agencies. Thank you very much.